In our last presentation, we talked about the history of genetics, the people who were involved in improving our understanding of genes, the structure of DNA and RNA. Now we're going to talk about how DNA and RNA are involved in making proteins. Proteins are a significant part of our body's organic matter. It's the same thing with microorganisms. That is about half of the organic matter in any living organisms, and it provides construction materials for tissues, for individual cells that play a vital role in cell functions. In individual cells, they act as enzymes. In larger organisms, they can act as hormones and antibodies. They also have structural and functional value within individual cells as gateways and for materials to move in and out of the cell, for instance. Proteins are made of amino acids. You really don't need to know the exact structure of an amino acid, but it does have a nitrogen group. And basically what proteins can do is they can be formed into a couple of different types of structures. There are fibrous proteins, structural proteins, that appear both in body structures and in cells. Examples for us would include collagen and keratin. These proteins are pretty stable. And then there are globular proteins, which are functional in nature. These function, again, in bodies as antibodies, but on a cellular level, they can function as enzymes, they can function as cellular pumps, they can function as part of any of those organelles that we mentioned. The problem with globular proteins is that they are fairly unstable, and they have to be in a pretty safe, consistent environment, or they can be denatured. By denatured, that just means that they can be unfolded in a way that leaves them non-functional. Example of denaturing: if you've ever cooked an egg, when you crack that egg open, it's runny, it's clear, but as you apply heat, it starts to change color and to thicken and solidify. That's because the proteins within that egg are unwinding. Now, proteins have multiple layers of structure. There is a primary structure, which is that amino acid sequence, which is controlled the DNA. Now, because of this primary structure, the amino acid sequence, the chain tends to get little twists or turns, or sometimes even has this kind of pleated sheet appearance. Beyond that, the proteins get folded, and as a result, they can take on a unique shape. That's the structure that we break apart when we denature them. Now, if we have two proteins that are non-functional separated but become functional put together, we have quaternary structure. And I think of this like having a fully functional truck up on concrete blocks. And what I have to do is I have to slap the tires back on the truck, and then it's functional. But until then, it is not. And that would be what I consider something of a quaternary structure. So this graphic shows you some of the levels of protein structure. Here's our primary sequence. Here is this twist that results as a product of that, and we can get that pleated sheet. The tertiary structure we don't really see here, nor do we see the quaternary structure in this particular graphic. Now, nucleic acids are part of DNA and RNA, and they provide the blueprint of life. DNA and RNA are made up of nucleotide bases. There are 5 nucleotides. Adenine, guanine, thymine, and cytosine are in DNA. Adenine, guanine, cytosine, and uracil are in RNA. So there is a slight structural difference. You should know then that DNA contains deoxyribose and the 4 amino acids just mentioned. RNA contains the sugar ribose and the other 4 just mentioned. Now, the structure that we were talking about when we talked about Watson and Crick, what they finally elucidated was that the structure of DNA is a double helix. That is, a helix is a twist, double because there are 2 bands that interact here. And they are formed by complementary pairing. We mentioned before that there was a discovery that adenine was always attached to thymine, guanine was always attached to cytosine, and this is what we call the complementary structure. Now, how does this play into protein synthesis? When we mention a gene, what we are talking about is a strand of DNA that can code for a specific protein. That is the blueprint for one protein. So a chromosome can have sections that code for hundreds or thousands of different proteins. Proteins have a lot of functions, like we mentioned before. They are building materials for the cell. They act as enzymes and biological catalysts. Now, RNA is also essential here, and that is ribonucleic acid. It is the intermediary between DNA and the final product of protein. There are 3 types of RNA that are involved. There is a messenger RNA, which is directly copied from the DNA. And then we have ribosomal RNA, which is a special kind of RNA that can read the message off of the messenger RNA and help build the protein. And it builds the protein with the assistance of transfer RNA, which is kind of like a gopher at a job site in that it goes and gets the particular amino acid that's needed. These processes are known as transcription and translation. And transcription is the transfer of information from the DNA sequence to the complementary sequence of messenger RNA. Now, it's that 3-base sequence on messenger RNA that's called a codon, and that codes for a specific amino acid in a protein. So, each codon codes for 1 amino acid. We have 20 amino acids, but there's only 4 nucleic acids. So the way this works is 1 nucleic acid in a codon would only allow for 4 amino acids. 4 nucleic acids times 4 nucleic acids would allow 16 if we had codons that were 2 in length. But 4 times 4 times 4 gives us 64 possibilities, which is plenty for all the amino acids, as well as codes for start and stop. Now, you don't have to remember all of these codes by any means. You don't have to remember any of them. This just demonstrates that there is some duplication. With 64 codes, we can have more than one code for one particular item. So, we have leucine, which is coded for by 4 different codons, or combinations of cytosine, uracil, uracil, cytosine, uracil, cytosine, cytosine, uracil, adenine, cytosine, uracil, guanine. So those are some of the possibilities that occur. Once that has been transcribed, that is written, we have to translate it, and translation is taking that base sequence of nucleic acids and converting it into a protein by bringing the appropriate amino acids, which are the building blocks of the proteins, into play. This process occurs, as you can see from this graphic. We have the nucleus, where we copy our messenger RNA off the DNA. Messenger RNA goes out, gets read by the ribosome, which builds a polypeptide using the transfer RNA to bring the appropriate amino acid to it in the process. So in summary, chromosomes are made of DNA. DNA is made of a combination of 4 nucleic acids. Each section or sections of DNA are called genes, which code for a protein. A gene consists of a long chain of nucleic acids that are in groups of three, and these groups of three are called codons, and each codon specifies one of the 20 amino acids that are used to build proteins. So to summarize that, DNA is copied to messenger RNA in the process of transcription. Messenger RNA leaves the nucleus, going into the cytoplasm where it is read in the process of translation. Translation occurs on ribosomes, which are made of protein and ribosomal RNA, and transfer RNA assists ribosomal RNA by bringing the correct amino acid to be added to the chain. That concludes this presentation on DNA, RNA, and protein synthesis. There are some adjunctives, again, off to the right-hand side of a soft chalk presentation, which I urge you to take a look at. I'm not going to have a third presentation on the Human Genome Project. What I would like you to do is to read that portion of your textbook starting on page 82 and through to the end of the chapter. Be sure that you know the definitions of the key terms, again, on page 86 of your book. And thank you very much. Hope this helps.